Hello, everyone. It's 8:02, so we're going to get started. Welcome to our audience here at the ATRF and to those joining us online. Thank you for joining our Spring 2024 seminar of the Biotech Connector Speaker Series. I am Maggie Scully, the Director of the Partnership Development Office here at the Frederick National Lab. The Biotech Connector event is made possible by a partnership between the Frederick National Lab, really spearheaded by Luba Kavutsky, and the partnership with Frederick County Chamber of Commerce. So thank you to all of those folks for supporting this event and making it such a staple here now in Frederick. The event is a quarterly series that brings together our local biotech community for an inside look of recent advances, a chance to share expertise, as well as an opportunity network. So I hope you all get to do that today with coffee. Here's a slide of just some of the contacts for relevant offices. The Frederick National Lab is a collaborative enterprise. We are interested in exploring partnering opportunities, and some of you may as well. We also engage in a lot of outside, engage with outside organizations through subcontracting, and those contract solicitations are available on our site, so please feel free to review that. We also are interested, if you're interested in career opportunities, we have some job openings posted on our site. So again, please review our website and feel free to reach out to the respective offices. Today we have three amazing speakers who are presenting on the topic of, electron microscopy. Just go through logistics first. We'll go back to back with speakers with time for questions in between. When prompted for questions, please stand up so everyone can hear you and state your question. For those online, please submit your questions in the chat box and we'll have someone read that out loud here, or if you prefer, unmute yourself to ask the question. Also, for those online, please remain muted during the event and turn off your camera for bandwidth, and note that the meeting is being recorded. Okay, now we'll get to the fun part of introducing our speakers. First, we'll have Dr. Ru-Ching. Hsia is the principal scientist and head of the Electron Microscopy Laboratory here at the Frederick National Lab for Cancer Research. Dr. Hsia has received her Ph.D. degree in microbiology and immunology from Stanford University. Before joining the Electron Microscopy Laboratory, she was a professor and director of the Electron Microscopy Core Imaging Facility at the University of Maryland, Baltimore, and in the Department of Embryology at the Carnegie Institute for Science in Baltimore. Dr. Hsia has extensive experience in transmission and scanning electron microscopy, light microscopy, instrumentation, and sample processing for biomedical research. Dr. Hsia is also the organizer and host of a bi-monthly webinar, BioEM talks, for those who are really interested in biological electron microscopy. Our second speaker is running late, so we will, hopefully he will be here on time, but it's Mr. Joseph Mowery. He is a biologist with a research background in electron and confocal microscopy. He is with ZEISS, but before that, he led the electron microscopy facility at the USDA, Agricultural Research Service, focusing on developing novel TEM and cryo, SEM, scanning electron microscopy techniques for various research applications. His work has contributed to over 70 peer-reviewed journal articles and is a recipient of the Hildegard H. Crawley Award from the Microscopy Society of America for his contributions in electron microscopy. Currently, he's a product and applications specialist for life science EM and X-ray microscopy at ZEISS Microscopy. Okay, and finally, our third speaker is Dr. Kedar Narayan. He's a senior scientist and group leader at the Center for Molecular Microscopy at the Frederick National Lab and NCI. Kedar has a PhD in immunology with an emphasis on biophysics and imaging, and a background in chemistry, pathology, and software engineering. Quite well-rounded, Kedar. His group has developed and applied focused ion beam scanning electron microscopy and other volume electron microscopy technologies to answer questions in cancer and cell biology. Specific areas of Kedar's research focus are correlative imaging, deep learning, artificial intelligence, and volume electron microscopy tool development. Okay, so now please welcome our first speaker, Dr. Hsia. Okay, thank you.
Thank you, Maggie, and thank you for coming early to listen to the talk today. So my name is Ru-Ching Hsia, and what Maggie didn't tell you is I just joined FNL in last September. So some of the talk that presented here, most of the work is not done here, as we are still trying to I'm still trying to settle in, and we do get some work started, but not ready to really present any data yet. So. My neighbor and friends often ask me, what do you do every day? So I always tell them, well, I'm looking for small, tiny little things in a very big instrument. And literally, I'm often looking for them because one of the biggest things, besides operating a big instrument, is preparing the samples. It's critical because EM samples has to be very tiny, and you are dealing with delicate, small pieces of samples. And when they jump off my hand, I'm literally on the floor looking for them all the time. Okay, so here is listed the major difference between electron microscope is the resolution. We stand up, there's no comparison that when you need to reach the resolution, electron microscope is the only way that you can view it. Like microscope, even with the super resolution microscope, they are still falling short for some of the resolution we needed to study some of the biomedical research. So electron microscope was invented in 1930s. This is a picture of one of the original microscope that's designed by Max Knorr and Ernst Ruska. Okay, and so since the electron microscope used the electron as the light source to image, so there's quite a lot of limitation for what you can put it in. So it took quite many years. Scientists like Keith Potter, Albert Claude, and Ernest Fulan, they took them quite a few years and publishing how you can put image cells in electron microscope. This is one of the first publication that in 1944 shows the first cell image in the electron microscope. You can see very poorly at that time, okay, but they've already developed using osmication to stain the cells, okay? So since then that we know electron microscope has contributed to the major understanding of cell biology. They discovered and described organelle like Golgi, ER, microvilli, and mitochondria. And these scientists was awarded Nobel Prize in 1974 for their discovery and description of a lot of the cell biology. That's what we understand to the present day. Okay, moving on. Now we actually, EM has improved a lot more. We push the resolution and push the functionality of the electron microscope that we can actually I don't think this place in the relay. Maybe how can I need to take this pointer off, right? Yeah. Yeah. Okay, sorry about this. Okay, so this is actually a short film that I often use in my presentation. So this film is made by Dr. Drew Berry, that he actually used all the real structure that's discovered in cryo-EM and X-ray crystallography. And to generate this film, I think taking like 2 years to generate this 2-minute film and following exactly the structure as described here. So this film actually depict the central dogma of protein synthesis. You can see the tRNA bringing the new amino acid and then going through the ribosome to generate the protein peptide chain. Okay, so I like to show these images these pictures because it reminds me 4 major things when I, as a cell biologist, using EM to study cell biology, function and processes. First thing, most of the functions, all the function inside the cells in our body stays in an aqueous environment. It's not in a dry environment. And second, most of the time the molecules don't act alone. When they function, they often function together, forming a complex of multiple factors. So that also reminds me that when we study just one molecule, we need to know they never really work by itself. It's often forming like a molecular machines, and they function together. The third thing it tells me is it's a dynamic event. So it's constantly changing, Okay, so we need to capture the most critical moment when we try to do imaging. And so now we are actually studying the most representative state of the process. And lastly, it reminds me again and again is things happen in 3D. So when we look at the 2D images, we need to know there are things, they are not just 2-dimensional. You have to really think of the real structure as 3-dimensional. Okay, so before I move on and also set the stage for the following talks, there's two principal electron microscope. One is called transmission electron microscope, and one is called the scanning electron microscope. Okay, so these two figures is describing the differences in the most simplified terms. Okay, so transmission electron microscope studies the transmitted, use the transmitted images. So your sample has to be, is blocking the electron. So then that shows the dark particle and dark imaging on the screen. And for scanning electron microscope, instead of collecting transmitted electron, we're collecting the electron bounced off the surface. So you actually study the topography of the surface and actually give you some 3-dimensional information. Okay, so because of transmission electron microscope need to transmit through the sample, so they need higher voltage. You can see it often has a much bigger column than standing in a standard SEM. Okay, so the bottom shows you the pictures of the same sample when you put in the trans-EM, how they look different. So basically, transmission electron microscope does not give you the internal structures, okay? But scanning electron microscope gives you a 3-dimensional information when you want to study the sample. So same thing here is a bacteria. You can see when we prepare the sample, put it in the TEM, you can actually see the internal structure of the bacteria. But when you study it in the SEM, it shows you the structure and outside and how they form and give you a volume information when you use SEM. So of course, these two instruments has been developed. There's many variations and things, and the boundary between TEM and SEM has merged quite a bit. You will hear it from the following talk today. Okay, so how does electron microscope advances biomedical research? So it's been heavily applied in most of the biology research here. So I'm just categorizing them into 4 major categories based on my work experience in the past and projecting the future, what is going to continue playing and contributing to our understanding of biomedical research here. So first is the nanotechnology, nanomedicine, and then the ultrastructure characterization. So even this characterization started in 1940s, we are still keep going. There's still new structures and new function we are studying and identifying from the EM. And temporal spatial analysis of molecular expression. So these are very critical as we are at this stage of trying to find new therapy target and new drugs and a new vaccine method. We are applying EM research allowed for us to move forward to understand the best way to design this new medicine. And 3D structure analysis are higher order. So this is going to be using scanning electron microscope that actually advancing to go to study a much larger order of cell organization instead of at the molecular level. So I'm going through them one by one and showing you some examples that we can do by using electron microscope. So the nanotechnology, and we hear nano a lot of times in this age when we do the biomedical research. So nanoparticles, there's all kinds of nanoparticles. So this picture actually shows a nanoparticle with a core, and you can coat it with biomolecules. You can load it with different things, and to do site-directed target, you can load different drugs or different using attachment function. So there's all kinds of nanoparticles that require EM to characterize. The next one is virus-like particles. We partner with cancer research. So there's a lot of HIV VLP comes in, people ask her to characterize. Again, people use virus particle to design as a potential target delivery mechanism and for drugs and also as vaccine target. So this specific picture is actually from a VLP produced. They are trying to use it's genetically manipulated, so they're expressing some target that they can use. So they are specifically asking us to look for loaded and empty capsid. So here you can see the loaded one are actually the one that looks more homogeneous, and the empty one that shows has a dark center. So you want to see more loaded vesicles when you want to get FDA approved to use it as a drug. So we do a lot of this type of work for people. And exosome, you probably hear a lot. Exosome was recently discovered probably 10, 20 years ago, that we found there's a lot of function that's actually, a lot of proteins actually secreted by cells using exosome. Okay, so all kinds of cells secrete exosome. That's including, you can isolate them from blood, isolate them from culture supernatant, and urine from patient, even plant cells has exosomes. So they are also called extra vesicles. So a lot of people are trying to take advantage to see exosome can be used for therapeutic method. And so we get a lot of sample, we're helping us to characterize. So you can see it's quite heterogeneous when you isolate things from urine or blood sample. You have to characterize them to separate them, which ones are exosome and which one are just some other particles. And liposome is also important factors. A lot of drug delivery is using liposome because they can fuse with cell membrane, can deliver things into cells. So a lot of studies using liposome, and again, they rely on electron microscope to characterize these nanoparticles. High resolution ultrastructure characterization. I said this is still 80% of our work. People are asking us to characterize ultrastructure. Even it started in 1940s, we are still following majorly the same protocol, prepare the sample to do the ultrastructure study. So I'm listing just a few topics that I was involved. I mean, the list can go more and more, but I'm just listing some of the important things that we have done and can be done, studying metabolic diseases, metabolic disorders. And so here is a picture showing the lipid droplet. It's an obesity study that we're trying to see how does lipid droplet biogenesis involve with obesity. And here is lipid droplet accumulation in a mutant in a heart muscle. You can see the large increase of the lipid droplet when we study that. And these lipid droplets are quite small, so you cannot really study them using light microscopes. You have to use electron microscope. Mitophagy. So many cell disease or dysfunction is manifested in mitochondria because this is one of the most important organelles in our cells. And so you can see in this picture, it shows some typical steps of mitochondria going through degradation. Okay, so you can see the degradation of Christie, okay, and you can see vacuole gradually starting forming, and you can see the accumulation of things in these, some of them called multivascular bodies. That's an accumulation of degraded organelles. So you see multiple membrane that's left because that's the lysis. So if this mitophagy keep going, you're going to see more mitochondria is completely like an empty vacuole because everything, the Christians and the mitochondria content is completely lysed. So a lot of characterization to help people to understand how does the drug work, how does the disease progress, and by looking at the mitochondria function. Infectious disease, again, microorganisms, you have to use microscope to study them. So here is a lot of work have been done trying to define how the interaction between microbes and the cells. So this is one of the study that's involved with the invasion of bacteria. Some of the bacteria can actually survive in cells. They specifically want to bind and then enter cells, actually help them to invade the immune defense. So bacteria use various strategies to cause disease. And so again, we have to use electron microscope to study the interaction. Finally, this is the work I was involved with Carnegie. We actually worked with a lot of ecology related topics. So one of the major focus is symbiosis and microbiome. So as you you'll probably hear about the coral reef that bleaching, and that is caused directly by the corals releasing the algae. So which are these dark particles when the temperature goes too high. The first thing they do, they release the algae, and algae actually functions as an energy source. They produce to help the coral reef grow. So when they lose algae, they start bleaching and dying. So there is a lot of study trying to understand the relationship between algae and the coral reef. And we are trying to set up in vitro models and trying to find out what are the molecules involved with this symbiotic interaction. So when we look at the larger picture of ultrastructure changes, the next thing we want to know is what are the molecules involved. So this is the next step. We're trying to actually identify the molecule that's involved, and that helps us to identify the strategy to help to either cure the disease or prevent the disease functioning. And so in this, we use a technique called immuno-EM. So immuno-EM, there are several methods you can try, and they all have pros and cons, and you have to really look at the specimen you are looking at, the target you are working with to design the best way to approach this. Basically, we use gold particle instead of fluorescence target marker to look at the molecule. So here is one of the bacteria that's labeled with LPS. You can see this labeling outside of the bacteria. And the next one is using a pre-embedding immunogold labeling. We found out the location of this specific target protein. It's between the muscle fibers. And all these work that help us to know where the molecule is, when they are expressed. And sometimes you can even do double labeling to see the interaction of this molecule with other molecules. And so the next one is called the Tokuyasu labeling or unembedded high ultra-thin section. As I emphasized earlier, most of the biological function, it happens in aqueous environment. So when you dry it, and you may destroy the molecule, destroy the interaction. So this is a method you actually freeze your sample in cryo condition, and you actually can label them when they are in aqueous hydrated state. So we also apply that to study some really sensitive molecules. And lastly, we are using correlative LN-EM approach. As I said earlier, fluorescence does not have the resolution EM can reach, but it does have the advantage that it can handle much larger pieces of tissues samples. So that's why the claim has been a really major push in the field, because this technique bridges the advantage of both EM and LM. So you can use a fluorescence marker and to locate this particle, and we actually can get the structural information, because if you know fluorescence images, you can only see the labeled target. You don't see the structure. So by combining these two, LN and EM, you can actually see the target and also locate the specific structure. So in this study, we are studying a Toxoplasma parasitophorus vacuoles. So they found the punctate expression of a host secretory target protein and inside the parasito vacuole. So they come to ask me, okay, we want to see what's going on here. So at the end, we were able to find out that Toxoplasma actually hijack the secretory function in the cells and use that to secrete nutrient for them into the vacuole when they are causing the infection. Lastly, the scanning electron microscope. Okay, so as I said earlier, scanning electron microscope gave us the volume information. Sometimes after you look at the structure, you find out the molecule, there are some implementations of the disease actually as happening at a much higher order of the structure. So here, the first image is a hair cell. So hair cell is the mechanotransducing receptors in our inner ears. Okay, so you can see they have orderly structure formed here. And so, but when we lose our hearing or damage these hairs, and or when people have Deaf syndrome when they are born, we often find the mutation happen at the hair cell, and it's implemented, manifested right at the structure. You can see the length of the hair cell reduced, and the structure order is completely destroyed. You can see them collapsing, and in the in the mutation. So again, scientists trying to dissect the mechanism, the cause of these hair cell damages and the molecule that's involved. So there's a lot of interesting work that's being done. So here is a mouse inner cell specimen that we did in the SCM. Okay, and biofilm is also a very important involving our daily life. So as I mentioned, you know, even bacteria, they often stay in a planktonic state in nature. So they often form groups. And when they form groups, they form biofilm. And biofilm is more resistant to drugs, more resistant to environmental factors. But it's a very delicate structure. Okay, if I dehydrate it, often this structure is destroyed. So, and often during in nature, there's a mixed population of bacteria. So this is why we use cryo-SCM. Fortunately, I have a cryo-SCM at University of Maryland, Baltimore. I called it my best kept secret because not many people have cryo-SCM. But cryo-SCM allow us to study a lot of things, has to stay in hydrated condition when we study the structure. So biofilm is one of them, okay? Hydrogel, that's also very popular right now. With the stem cell research, people are trying to do tissue engineering and design what's the best scaffolding material. So hydrogel is very hydrated material. If you dehydrate it, all the structure is going to be destroyed. So then we use cryo-SCM in order to study the structure, the mesh network, and the pore size of the hydrogel. And people are also trying to load growing stem cells with the hydrogel material. And yes?
Do you lose the resolution when you do 3D? Because in the previous slide, the scale bars and nanometers, here it changed to microns. So do you lose that spatial resolution?
Well, these are just larger the holes and all that. Yeah, ACM does have less poor resolution comparing to TEM, but the nature of these samples are larger sizes here. So that is also why some of them is difficult to study in TEM, because TEM has a very limited size that you can put it in. Sample has to be very thin, very small. So often you won't be able to put a whole hair cell structure to study in the TEM like this. Okay, yeah. So coming back, you can see the stem cells embedded in this hydrogel here, scaffolding material. Again, this is a study has to be done in cryo-SCM because you cannot do it in TEM when they are dehydrated. And also in order to study these larger pieces of hydrogel with multiple stem cells embedded, you want to see the interaction, how the stem cell grow and if they spread and in the hydrogel. So this is some of the study I am involved. So I quickly go through some of the topic I involved with BioEM research using electron microscope, but it certainly does not cover everything. And there are still quite a lot of limitations for EM work. And one of the things is instruments are costly and expensive to maintain. So all these bad boys, they can cost millions or thousands, hundreds of thousands of dollars, and it's difficult to maintain. So not many research institutes have all the instruments, and the sample preparation is tedious and labor intensive. As I said, my major work every day is not looking at the specimen in microscope. It's actually preparing the sample because it is tedious, and you have to know which is the best method to prepare the sample and to put it in the EM. So because of that, it's difficult to do high throughput. So we are lagging behind with the genomic and proteomic studies when they can study thousands of samples, and we are still doing 10, 12 samples every time, but we are moving toward automation. We recently got the robotic platform. We're trying to really push forward to increase the productivity and efficiency. And lastly, the skills are not easily acquired. As I don't know if you know, many colleges do not offer electron microscope courses. And it's a difficult skill to acquire because it takes a lot of practice. And that's also contribute the slow growth in the field because there's less people here. But that's why we are here. Electron Microscopy Laboratory is here to help people to do research. So we do sample processing and imaging, but we also do consultation, really to help people to design the best workflow and sample preparation. We also help people with data analysis. And I do offer training opportunities. And as Maggie mentioned earlier, I run a bi-monthly webinar. It's free, called BioEM Talks. We have more than 600 subscribers following, and we discuss biological related topics. So if you are interested, just sign it up, and then you can join at any time. I'm going to stop here and take any questions if you have. Yeah. So far, no, because we do get quite a lot of server storage that offered to us here. So we don't, most of our projects are kind of small existing, and then once we finish it, we move on to the user. So I don't really have to store them for a long time because the data is owned really by the client. So has not been a big problem for us. Yes? Yes, EML, we do have our own microscope. That's because it's difficult to mix room temperature work with the cryo work, because once the instrument is cooled down, and it's difficult to put it back to room temperature, you have to bake it and all the stuff. So we kind of separate the cryo work from the room temperature work. So we do have three. Yes, we have both of that, and then we do work. Yeah. Okay, thanks.
Thank you, everybody, for being here today. And thank you, Ru-Ching, for the very nice introduction into the basics of electron microscopy. My name is Kedar, and I'm a group leader here at Frederick National Lab. And what I'm going to be talking for the next few minutes is an approach called volume electron microscopy that some of you may have heard of and others may have not. So I'll go through some of the basics of what volume EM can do and why I think it's really transforming cell biology. Okay, quick takeaway. What do I want folks to get away from today's 15-minute presentation? Volume electron microscopy enables the three-dimensional visualization of cellular structures at nanoscale resolution. Right? So as Ru-Ching mentioned, at much higher resolution than light microscopy, except volume EM, unlike the images that you saw previously in our presentation, are three-dimensional. There's a host of hardware and software advances recently that now allow us to image larger features of interest. And when you have rare events that we call needle in the haystack problems, we can use correlative imaging that combine light and electron microscopy to image what we want. And a very interesting aspect of what we've been doing in lab is to create artificial intelligence-based approaches that target known bottlenecks in the volume EM pipeline, which are now changing the kinds of questions that you can ask with electron microscopy. So if there's nothing else that you understand from the next 12 minutes, it is that imaging cellular architectures in 3 dimensions and at high resolutions can now reveal biology that was previously unknown or previously inaccessible because of technological limitations. Okay, I'll very quickly go over a movie that reiterates what Ru-Ching spoke about. In transmission electron microscopy, you have an electron source at the top, you have a series of lenses that look like rings, and you have to put a very thin sample in order to shine the electron beam through it and get what we call a projection image. And that sample has to be 100 nanometers or so, which is, if you think about a cell, which is about 10 microns, represents about a 1% of the thickness of the volume, meaning that your image, here the lymphocyte, which is activated, yes, you get extremely good high-resolution images of the insides and the outsides of the cell, but it's a representative thin slice. Scanning electron microscopy, on the other hand, again, Ru-Ching showed a very nice schematic, Here you have the electron beams going again through a bunch of lenses, and now it hits a sample, and the electrons scatter off of the surface of the sample, and you can now either raster the beam or move the sample. But the point is, as the beam rolls over the sample, you can get a very nice image of the surface of the sample. So what you're looking at here is another lymphocyte, and now you get a three-dimensional looking image. But remember, this is still a 2D image, right? It just looks like it's 3 dimensions because for all these bits and bobs and nubbins on the surface, anything that protrudes scatters electrons efficiently. Anything that's recessed does not scatter electrons. So you get this idea of depth, even though this is just 2D. So where does volume electron microscopy come into this? This is the conventional scanning EM image that I just showed you, right? You get the surface of the entire cell, or there's transmission EM, where you get the insides and the outsides, but only of a 2D slice. Now there are hardware and software advances that allow you to get a TEM-like image in the SEM, right? So as long as you cut open a cell, you can now, in the scanning electron microscope, get really high quality images. So why do we care? The reason is because now TEM, you have to physically section each of your resin embedded samples, take it to the microscope, take a beautiful image. But with latest hardware and software developments, this process of slicing and imaging can now be automated, meaning that you can get a stack of images that goes through the entire volume of a cell. So rather than a representative 2D image, you get a stack of images that goes through the entire cell, meaning that you can get a 3-dimensional reconstruction of what's on the inside and what's on the outside. So the best of both worlds. And now what you can see here is I just false colored the nucleus purple, the mitochondria pink, and the plasma membrane of this T lymphocyte, the same T lymphocyte as it happens in 3D. So that's the advantage of volume electron microscopy. So instead of getting representative 2D images, you can get the entirety. Important clarification, you may have heard of cryo-EM. This is the technique that won the Nobel Prize several years ago. Cryo-EM looks at proteins at high resolution and in 3D. Volume EM looks at cells in high resolution and in 3D. And another distinction is that it's not just one approach. There is a series of related electron microscopy based approaches that can essentially give you images at slightly varying XYZ resolutions and are better suited for features that are differently sized. But note the y-axis is tens of nanometers, so still significantly higher than light microscopy. And in our lab, we have several of these microscopes. As Ru-Ching mentioned, these bad boys are quite expensive, and they shine at specific areas in this graph. So very small things, slightly larger things, and significantly larger things at high resolutions. Volume EM has recently had a big inflection point. A lot of advances have come together. So along with the James Webb Space Telescope, along with CRISPR, Nature last year touted volume EM as one of the 7 technologies to watch. And a few of us in the community wrote an in-depth primer, which is up there in the middle. And for those of you who just want a commentary, this came out in Nature Methods last year, just a 2-page summary of what volume EM is. I would urge you to read it. A few of us edited the first Methods in Cell Biology book if you're interested in more practical aspects of how a volume EM experiment can be done. And this is what I meant by the field is in the midst of an inflection. If you look in PubMed, the number of publications has just risen dramatically, although you see that the y-axis is significantly lower than traditional EM. So it's still a field that is in its nascence, but it's empowering cell biology. So the obvious question you can ask is, why do we care? Why do I go on and on about viewing things in 3 dimensions? Well, the answer is, even if you take the most simple 3-dimensional object, a cube, and if you take representative 2D slices, you can imagine that depending on your orientation of how you slice it, you can get dramatically different looking 2D images. It's the same cube, but your 2D images look vastly different. And if that is true for a cube, you can imagine for complex biological structures like mitochondria, they only look like kidney beans in textbooks. In real life, they can look dramatically complex, right? And that means that you need to truly understand what these organelles are, you need to image them at high resolution in their entirety and in 3D. And so that's what volume electron microscopy allows us to do. And in our lab, we are deeply interested in mitochondria. Everyone from high school and above knows that they are the powerhouse of the cells, right? And so mitochondria are ubiquitous, they're central, they're really important, and they show dysfunction in a variety of pathologies. So we are deeply interested in mitochondria. And I'm just going to give you one example of a project, a collaborative project where BioEM has really shown dramatically new readouts that were previously impossible to do. So the liver is made up of these repeating units called lobules. They're a cluster of cells that repeat over and over. And essentially what happens is, very, very basically, blood comes in through the portal vessel, traverses through these blood vessels, and then empties out into the central vein. And these pathways or channels, if you will, of blood are surrounded by liver cells or hepatocytes. And what was known is that not all hepatic mitochondria are the same. So depending on how proximal they are to the periportal vein where the blood comes in, or the central vein where the blood dumps out, depending on where they are, the hepatic mitochondria are different. So the liver lobule essentially has zones. And if you have various cell surface markers like Kedar or CD73, you can tell by light microscopy that these, at low resolution, that the lobules are divided into these zones. And in vitro studies have suggested that something called the redox potential, which is sort of a readout of how mitochondria work, they're also different in these zones. But the question that we wanted to ask is, are there any structural differences in these mitochondria? So we know, okay, there are functional differences that we know from in vitro studies, but if you get a biopsy in preclinical or clinical studies, you can't do in vitro assays. So now, can you tell if there are structural differences in these mitochondria that correspond to these functional differences? And so what we did was this is a part of the liver globule stained by toluene blue from a biopsy. We then prepared this for electron microscopy, and what you see now is about a millimeter chunk of the tissue that goes all the way from the central vein at the bottom to the portal vein at the top. And now if you take high resolution images of these in 2D, you can tell that the mitochondria generally look different, right? These mitochondria look different from these mitochondria. So up until now, what we would have done is traditional, descriptive, qualitative observations. And there's nothing wrong with this, because this is how, as Ru-Ching correctly pointed out, this has been a cornerstone of cell biological research, right? So figure 1A, for example, in traditional electron microscopy papers would show something like this. You'd have an arrowhead and you say, well, the periportal mitochondria are round, whereas the pericentral mitochondria tend to be long and skinny. Or you might have another arrow that shows, well, there are some mitochondria that are skinny in periportal and they're rounder in pericentral. And then you may have an asterisk for a feature of interest, or a lipid body. So these are a series of representative qualitative observations, which is fine. Nothing wrong with it, but these are, when they're manually acquired, it means that you are limited by how many N you can have, right? How many features you image. They're qualitative observations. They're not parameterized. So if something looks sick, what one EM person says is sick may not be what another person says is sick. And we know that the human eye is a pattern recognizing machine, which means that unintentionally, you may choose areas that are of interest, which can bias your readout. And finally, it's two-dimensional, which means you lack three-dimensional context. So our aim in our lab was to move away from this modality of search and snap, where an operator looks around for something interesting and takes a beautiful image, to something that's automated, computationally acquired, and analyzed. So in order to do this, we must image these mitochondria in 3D in their entirety. And then this is something that volume electron microscopy can do. Here's a quick movie to show you how we do it. I'm showing you this with adherent cells, but it's the same with tissue. You stain these with heavy metals, you fix them with aldehydes, you resin embed them like the mosquito in Jurassic Park, so they're embedded. And now what you do is you put it in the electron microscope. And the scanning electron beam creates a single image. And now a blade, which is inside the microscope, takes a very thin slice, a little bit like a nanoscale deli slicer, right? So you image, slice, and you build up a stack of images of these cells. Another way of doing the same slicing and imaging is by using an ion beam to do the slicing instead of a physical blade doing the slicing. But essentially, it's the same thing, right? Instead of one representative image, you get a stack of representative images, which you can then computationally combine to get a three-dimensional reconstruction of your cell or of your tissue. And so with volume EM, what you can then do is you can computationally extract out features of interest, like the mitochondria, to visualize and analyze them. So instead of 2D now, if you were to look at these periportal and pericentral mitochondria, here's what you would see. Instead of a representative 2D image, you get a 3D volume of these areas. And finally, you could still say, well, it's just a 3D image, so what? How am I doing for time, Tuba? I got 2 minutes? Okay. There's a big difference between fluorescence and electron microscopy. Yes, fluorescence microscopy is low resolution, but there's another important difference. With fluorescence, you see what you're looking for. If you want to look at nuclei, stain for DAPI. If you want to stain for mitochondria, you stain for mitotracker. Whereas with electron microscopy, you get the ultrastructure of everything, right? And that's a good thing or a bad, and a bad thing. So you can make serendipitous discoveries, but the extraction of your features of interest is a lot more challenging. If you have a massive amount of tissue with light microscopy, you just stain it with mitotracker, you'll only image mitochondria. Whereas this has to be done intentionally with electron microscopy, and this is a huge bottleneck in the field. It's very difficult to do manually, and this is the rate limiting step. So even with those of you who have never seen mitochondria in EM before, if I were to tell you, well, you can clearly see that there are 2 mitochondria here. Humans can do this very well. We knew that when we want to do it at scale, we have to use artificial intelligence. But it turns out that current algorithms suck, right? They don't do a very good job. So automation is the only way to do segmentation at scale to recognize mitochondria in these EM images. But for a variety of reasons, it was very challenging. And what I'm going to show you is a software plugin that we have created and released. It's open source, which allows us to do this at scale. Here are some fancy graphs just to show you that we are currently the state of the art, which means that our model is the best around. And that this was published earlier last year. And this is how science research goes, right? That's 4 years of work, 3 papers in 1 slide. But take my word for it, this is how Empanada works. If you have a volume EM data set, you open it, and your task, remember, is to identify each and every mitochondrion. Here's what that stack of images look like. Here's a whole host of mitochondria. Essentially now with our software, you go click and hit run, and you can segment each of the mitochondria automatically. This is in real time. This is not sped up. And just to show you what that does, you can get about 50,000, 60,000 mitochondria in about 4 hours. An intern was able to do 200 mitochondria in 1 month, right? So 200 mitochondria in 1 month manually, tens of thousands in hours. So that's what AI can do. So going back to the issue of the liver, what we can now do is you can take that entire volume that I showed you, apply Empanada, and now automatically, at a click of a button, you can get each and every mitochondrion in 3 dimensions. And so what that allows us to do now is go from representative 2D images to 3D reconstructions, and finally now you have 3D reconstructions, you can do quantitation. You can get statistically significant differences in things like volume, sphericity, and so on, meaning that now you can say you have quantitative 3D characterization of features by volume electron microscopy. So this is the paradigmatic change that we think volume electron microscopy plus artificial intelligence can bring to cell biology. And in unpublished data, which is very, very recent, what I showed you was a representative cell, right? So using Empanada, you can say the mitochondria are done in purple, in nuclei in purple, mitochondria in green, lamellar bodies in yellow. We can do it now not just for one cell, but for massive amounts of tissue. So this is the kind of AI, and by the way, all of this is not fluorescence microscopy. This is electron microscopy at 10 nanometer resolution. So volume EM allows you to image cells and tissues at nanoscale resolutions in 3D. We're really focused on automation and computation and AI-based approaches. It is excellent for high-resolution deep dive characterization, and now we're beginning to get quantitative readouts. It is still not good for high-throughput screening. It will never replace high-throughput fluorescence microscopy. Like most things, you have a Bermuda triangle of resolution, field of view, and time. You can pick two, you can't have all three. And essentially, we think that volume EM and AI can now rediscover cell biology in ways that was previously inaccessible. And so with that, I'll stop, thank all the folks in my team and our high school annotators, and stop for questions. Yes.
Is empanada trained specifically for mitochondria, or can you apply that?
Very good question. So first of all, it's open source, so feel free to download it and use it. Empanada allows you to run inference on any model. So when we release Empanada, it comes prepackaged with a deep learning model called MitoNet, which is specifically for mitochondria. But what I showed you in that very last movie with the zoom out, we now have NucleoNet, we now have, LBNet and so on. So you can use the Empanada framework to create and fine tune your own models for various other features of interest. Although please cite us if you're doing it, or feel free to collaborate with us. Yes, Tommy.
Or is there a way that you can use some sort of tool, that would create some density on a desired structure that would make it easier to sort of segment or identify?
Yes. So there are approaches. There's benzidine, for example, DAB staining, which further enhances the heavy metal staining, which makes in a targeted kind of way. And that allows easy segmentation. But again, that requires genetic manipulation, right? You need to have a tag, et cetera, and it's possible and other people can do it, but my aim really is to have this go in the direction where it is ready for biopsies coming from preclinical trials, for example, where you don't have the opportunity to do genetic manipulation.
I guess the follow-up question would be, if you're looking for a particular molecular complex.
Yes.
Or something like that.
I see. Okay, yes, that is possible. However, in order to get the resolution to be able to get those protein complexes, you have to make severe compromises on how much you can image or how long it'll take. So that's something that with correlative microscopy is doable, but it's more for niche applications, perhaps not for large volume imaging.
How thin are the slices?
The slices are approximately 10 nanometers to 15 nanometers thick. So the XY resolution is anywhere between 5 and 15 nanometers. And the Z slices, we have shown we can go down as much as 3 nanometers, but we typically image at about 15 nanometers.
And is there a size limit to the sample?
Depends on the technique. So that's the graph, right? So if you have very large samples, there are certain approaches that are not conducive. So you will have to compromise a little bit on resolution, but now you can image about 1 millimeter, which, by the way, for electron microscopy it's a terabyte data set. So these are vast amounts of data, but fortunately we have great computational support. Again, depends on acquisition parameters, but at high resolution, you're looking at a handful of mammalian cells a day in automated overnight runs, or about 1 millimeter, several sections of millimeters in a day. Yeah. Kendra.
It's your AI model to other types of volume data. So like if I wanted to use your MitoNet on say up to milligram with mitochondria, would it work?
It does. Very good question. The model is trained on every publicly available volume EM data set that has ever been published until November 2021.
But all fixed.
But all fixed and not cryo. So we made a strategic decision that the model was really getting confused. So it's all room temperature work. Cryo will be when we get one more computational person in the lab.
Okay.
Okay. Oh, sorry. Yeah.
I know we're not supposed to take pictures, but can you go back to the slide with the QR code?
Yes. Yes. Oh, yes. Please, please do take a picture of this. And for those of you who want to download it, I promise you the documentation is gold standard documentation. We've had high school kids being able to install Empanada and use it. So we are very proud of the documentation. Everybody good? Okay, thank you.
Okay. It's not coming up. George, I'm not sure, it's not coming up. Just a second. Not sure. Okay, he's coming. Sorry, I don't know if that's stuck. It was over here, but. Apologize for that. I guess it fell asleep. All right. Thank you.
Hello, everyone. Thank you for sticking around. I'll try to keep this short, maybe another 10 minutes or so. So my name is Joe Mowery. I'm the BioEM specialist for ZEISS. I don't want to duplicate too much of what Kedar and Ru-Ching did such an excellent job giving you an intro to EM and volume EM, but perhaps we can treat this more as a summary. So of course at ZEISS, we have a large portfolio of wide range of instruments from light to electron microscopy, and we'll talk a little bit more about our SEMs. So the great thing about SEMs, of course, is they're really flexible, so you can add a lot of different detectors on them. So you have a lot of flexibility compared to traditional TEM to collect a lot more different types of electrons and signals. Particularly, we're interested primarily in secondary electrons and backscatter electrons and potentially some transmitted electrons. So again, I'll review some of these techniques here. Just kind of go through and kind of really nail home all the different techniques that you have available for SEM. So starting off, have some. Okay, 1 second. So I guess the most traditional type of SEM imaging would be was more topographical based. So you're looking at 3-dimensional specimens, and let's see if this is going to work. Nope. Okay, so this is great technique for looking at things that are very 3-dimensional, cells, tissues, bacteria, for example. Of course, you have to do a lot of work to get them to this stage, because you can't just put biological tissue inside a vacuum chamber, right? All the water and moisture will get pulled out of that, and the tissue will shrink and shrivel. So you have to fix things, dehydrate them, and even coat them into metal. But if you do that properly with the right protocols, you can get some great images. Another technique and of course, we're not just doing single images, but we can tile and stitch. Now that we have access to digital cameras, we can, capture a large field of view. So this is almost 3,000 images tiled and stitched together. This happens to be a monkey brain that was kind of prepared to reveal the blood vessels. Now, another technique here is called variable pressure mode. So this would be basically introducing a small amount of nitrogen inside the chamber and then essentially lowering the vacuum. So that allows you to then, depending on your samples, maybe you can put your fresh tissue directly into the SEM without doing those chemical steps. You can also get around coating in some cases. But you're going to sacrifice a little bit of your image quality because now you have gas that's interacting with, interfering with your detectors and things like that. But it's a really great technique if you're just wanting to get an image really quick without some of the sample prep. Okay. Moving on to cryo-SEM. What is cryo-SEM? This would be kind of for, I would say, now how do you get around, how do you improve the image, right, without doing those chemical processing steps again. So you have this ability to, with cryo, is to get really nice lifelike preservation. Instead of when you're chemically fixing things, you can generate a lot of artifacts, which is really not what you want. So by doing cryo, you basically freeze your tissue. You keep your sample the whole time on a cryo stage for all the imaging. And so you can get also images like this, where this is chemically fixed and didn't really turn out that great because it's all dehydrated. But when you compare that to what you can get in a SEM cryo, you can get much more lifelike image. And in this case, you're actually capturing more of the behavior, and you can see this mite is actually feeding. So this is actually transferring a plant virus in Mexico that's now invading the US here. Now, so those are kind of the three techniques. So like standard SEM imaging, variable pressure, and cryo for kind of topographical 3D imaging of single specimens. But the rest of the techniques will be more focused on tissue and looking at sections. So the most basic of these is scanning transmission electron microscope. So also, similar to what you can do in a TEM, you can do in a SEM, and you can get, at high energy, you can get really great images of membranes and virus particles all in an SEM. The big limitation here is that your grids, which you're putting these sections on, is only about 3 millimeters. So those grids are also very fragile. So if you're wanting to now look at more sections or look at larger tissues, you need another technique. And that is where backscattered electron detection comes in. So in this case, you can get the same images, the same type of images, but now you can put those directly onto, let's say, a cover slip or a silicon wafer, for example, and you can see, you can put a lot more sections, you can put them wider sections, and yeah, a lot more, and it's going to be a lot more robust, less delicate when you're handling it. And you're still getting, great resolution. You can see synapses and yeah, a lot of mitochondria. So now, so this is still basically 2D images, snapshots, representative images. And now let's talk more about volume imaging, going into what Kedar was talking about. The first technique here would be array tomography. And this basically gives you thin slices or thin volumes. And in this case, we've correlated it with some confocal data and SEM to give you a nice correlative aspect which is really helpful. But the limitation here is that your volume is relatively thin. The nice thing is you can do the qualitative aspect and you can get really wide fields of view. But now if you want to now increase your volume, that's where you need a different technique called serial block face imaging. And I happen to have a nice video here that kind of shows you clearly what that is. So you take a standard SEM, you remove the chamber door, you put on this other serial block face microtome door. This contains the entire microtome. And if you zoom in here, you can see the sample. It's about a 1 millimeter cube on a pin. And you have a diamond knife inside the chamber. So basically, you can now cut very thin slices up to 25 nanometers, maybe pushing it a little further to 10 nanometer thickness, and generating a backscattered image, and basically iteratively cutting and imaging until you generate a volume. One thing that happens, of course, with anytime you're imaging resin tissue, you get this electrical charging building up. And so we've developed this charge compensation system to mitigate that. Basically, what we're doing is streaming a small stream of nitrogen gas across the surface of the block directly before we image it. Here you can see where you're able to tile and stitch and select different areas of interest within the entire volume there. Here you can see where it's actually imaging. And you can see it's we're not actually collecting the slices, but we're just basically scraping them off. So that's how we can get really thin. And of course, we can get really large volumes at this point in a really automated fashion. And we're not just generating that volume to have it or to look at it. We're now going to segment that data and do some actual quantitative statistical analysis. So that's serial block phase. Now, which is a really great technique for large areas, tissue level, research. If you're wanting now to look at single cells or maybe map the mitochondrial network inside a cell, then you might want to explore FibSim. So FibSim you'll have a lot thinner resolution, somewhere around 5 nanometers, like Kedar said. And this is really focused on, single cells, and you can see the real detail that you get. The nice thing about this is because we have the ability to do like 5 or 3 nanometer slices, that means the resolution is isotropic. That means in any direction that you slice this, it will be, nicely uniform. So you can have the flexibility to kind of slice and examine the data from any angle without kind of stretching and warping. And you can see the mitochondria, vesicles, everything. And of course, you're not just doing that for the fun of it. You need to then do something with that data. And so you're going to segment out a lot of the different structures using AI and machine learning to get something that looks like this, and then do some statistical analysis. And so you can see here different vesicles. This is a nuclear membrane. Some mitochondria, etc. So this is focused ion beam SEM. Now imagine if we wanted to combine two different techniques, the cryo aspect with FibSim, then you get cryo-FibSim. So this would be a slightly different technique where you're milling and imaging into frozen cells. You don't have to do that fixation and embedding protocol, and you can create, using the ion mill, create a volume and mill into your cells. We also have fluorescence that you can correlate with, and then you put this into a cryo-TEM to do this tilt series tomogram, and basically the end result is in this case, would be a reconstruction of a protein. So that's cryo-FibSim. And now one last technique here would be multi-beam SEM. So everything up to now has been single electron beam. What if we had the ability to add more beams? Then we get a technique called multi-beam SEM. And this is really kind of at the moment, rather niche kind of technique, really being used for brain mapping, primarily in connectomics. But you can see here, we take the single beam, we split it using some apertures, and now we're imaging with multiple beams all at once. So this enables us to image large areas very, very quickly for high throughput. And this is, I think, showing you 9, but in actuality, it's up to 91. So it looks more like this. So this is really kind of pioneered by Harvard, Genelia, Princeton, and those folks are doing a lot of connectomics and neuroscience. And so you can see your images here getting generated simultaneously. So you compare that to what you would get with a single electron beam versus 91. Of course, then you have to handle the data, and so you need partnerships with Google and other ways of handling that large amount of data. So I'll keep it there. Just a quick summary of the techniques that you have available to you. And most of these techniques are available here as well. So thank you.
Yeah. So when you compare like a HeLa cell, the embedded technique to the Cray technique, do you see vast differences in the ultrastructure? I mean, is the embedding technique creating some artifacts, or is it kind of, are there, is it like within the ballpark?
Yeah, I'd say it's within the ballpark because with standard FibSim and embedding, you're embedding so much metal that you are really preserving it really well. Cryo can be really difficult because you then don't have any of those stains, and so getting contrast in those structures to pop out can be challenging, and of course your field of view is much smaller. Yeah.
The serial block, you're in a vacuum and you're slicing kind of 1 millimeter squares off. Where are they going?
Yeah. So yeah, or 25. Yeah, 25 nanometer. But yeah, they basically they're statically charged, so they stick to the metal around the block, and they'll pile up onto the diamond knife, and then so. But usually not a problem. I would say after you have that imaging for a day, maybe you have 1,000 sections, it might be good to open the chamber, brush some of that off. Okay, thank you.
Thank you so much. I know we're over time, so I would like to, thank our amazing speakers. Thank you, Joe, for making this through all this traffic and acknowledge and great thank you to our co-organizers, Frederick County Chamber of Commerce, and our amazing sponsors, Mike Bimo and Vanik IP for the breakfast. So the next event, Biotech Connector event, is coming August 22nd. Hopefully everyone will come back refreshed and ready to listen to, more science then. So thank you so much. Grab some coffee, and if you have time, you can chat with the speakers or, to each other. Thank you.